think we are discussing about GM crops, na? BT cotton, BT brinjal over. Okay. Next, STBT cotton. STBT cotton. Already BT cotton. BT cotton means Bacillus thuringiensis cotton. In that, the BT gene called Cry1AC gene have been introduced. So which acts like an insecticide or pesticide. So in that, another gene which is herbicide tolerant gene. Herbicide tolerant BT cotton. STBT cotton stands for herbicide tolerant, or you can say herbicide resistant BT cotton. So already the in BT cotton, the gene have been introduced which is acting as an insecticide. In a similar manner, another gene have been introduced which is resistant to the herbicides, or you can say weedicides. Here one gene called CP4 EPSPS. CP4 EPSPS. CP4 EPSPS gene have been introduced from a soil bacterium called Agrobacterium tumefaciens. Just to remember this one. Into this BT cotton, a gene called CP4 EPSPS have been introduced from soil bacterium called Agrobacterium tumefaciens. Which makes this plant herbicide resistant. Right? Just you remember this plant. That's enough. STBT cotton. Already BT cotton insecticide gene is present, and another gene called CPSP4 has been introduced from Agrobacterium tumefaciens. Thereby, it can act like an herbicide resistant. Okay, right. Another one, Arka Subha. Arka Shubha. The scientist from Indian Institute of Horticultural Research. Indian Institute of Horticultural Research, IIHR. So it is in Bengaluru. The scientists from Indian Institute of Horticultural Research have developed a new marigold variety. It was named as Arka Shubha. New marigold variety which was named as Arka Shubha. What is the importance of this Arka Shubha? Indian Institute of Horticultural Research. So what is the importance of this Arka Shubha? So this Arka Shubha has a keratin content of nearly 2.8%, which is the highest of all the plant varieties having beta carotene. Normally, the normal marigold is having a beta carotene of only 1.4%. 1.4% beta carotene is present in normal marigold variety. But in this Arka Subha, the carotene content is of nearly 2.8%, that is double the normal value, which is the highest of all the plant varieties which carries beta carotene. Okay, and that too after spoilage. We cannot extract this beta carotene from the normal marigold, but from this Arka Subha, the beta carotene can be extracted after spoilage. After spoilage, that is also important. So the carotene content is 2.8%, and that too the carotene can be extracted from this Arka Subha after spoilage, even after spoilage. Write down like this. The carotene can be extracted. The carotene can be extracted from Arka Subha. From Arka Subha, even after its spoilage. Even after its spoilage. Right. Next. The next one is transgenic mustard or GM mustard. Transgenic mustard or GM mustard. So there is a mustard variety called Brassica napus. Brassica napus. So this is a mustard plant. Into this mustard, the hirudin gene is introduced into this mustard plant, that is Brassica napus. The hirudin gene is introduced, thereby it becomes transgenic mustard. So from this Brassica napus, so hirudin gene is introduced from the seeds. The seeds are collected from this Brassica napus. From this seed, the hirudin protein is extracted. Hirudin protein is extracted. So what is that hirudin protein? What is hirudin? Hirudin is, it is an anticoagulant. Hirudin, it is an anticoagulant. Released by leech. Hirudinuria. So leech means jalaga. Okay. So what is this hirudin? The hirudin is a protein which is released by leech. Actually, leech is a sanguivorous animal. Sanguivorous means it's a blood sucking animal. Okay. So during the action of blood sucking, it releases a protein called hirudin, which is acting as an anticoagulant. That means it acts as anti-clotting factor. Now this hirudin protein is extracted from the leech, and from this hirudin protein, hirudin gene is developed in the laboratory, and this gene have been introduced into Brassica napus plant. And from this Brassica napus, the seeds will be collected. From these seeds, again the hirudin protein will be extracted, and it can be used as an injection, which is acting like an anticoagulant. Got it? Arthamainda? Right. So leech undi kada jalaga nunchi anticoagulant hirudin అనేది release అవుతుంది. Okay, this hirudin gene is constructed in the laboratory, and this gene can be introduced into the Brassica napus. Avalu antam kada, Brassica napus means mustard, avalu. So into this plant, the hirudin gene is introduced. Okay, it accumulates in the seeds of this Brassica plant, and from these seeds, the hirudin protein is collected, which is used as an anticoagulant. ఈ హిరుడిన్ ప్రోటీన్ని కలెక్ట్ చేసుకొని దాన్ని ప్యూరిఫై చేసి దాన్ని మనం ఇంజెక్షన్స్గా వాడతాం. ఎక్కడైనా క్లాటింగ్స్ ఉన్నాయనుకోండి, బ్లడ్ క్లాట్స్, ఆ క్లాట్స్ని కరిగించడానికి కోసం యూజ్ చేస్తారు. గట్ ఇట్? లైక్ దిస్ ఈజ్ ట్రాన్జెనిక్ మస్టర్డ్ ఆర్ జిఎం మస్టర్డ్. అండ్ అనదర్ వన్ ఈజ్, అనదర్ జిఎం క్రాప్ ఈజ్ DMH11. DMH11, this is important. DMH11. DMH means Dhara Mustard Hybrid 11. Dhara Mustard Hybrid 11. DMH11. Normally, the mustard plant is mustard. This mustard plant is self-pollinating crop. The mustard plant is a self-pollinating crop. As it is a self-pollinating crop, we cannot produce hybrids from this crop. Okay, as it is self-pollinating. Self-pollinating means that means suppose say these are the pollen grains. Okay, so the pollen grains released from this anther reaches to the stigma of the same flower. This is pollination. Transfer of pollen grains. Pollen grains means male sex cells in plants. So the transfer of these pollen grains from anther to the stigma of the same flower, that is called as self-pollination. Okay, transfer of pollen grains to stigma of different flower, it is cross-pollination. As it is a self-pollinating crop, we cannot produce hybrid varieties in this mustard crop. Got it? In order to develop hybrid varieties in the mustard crop, scientists have genetically modified these mustard varieties. After that, they have been cross-pollinated. Thereby, they got a new variety. That new variety was named as DMH11. Right? In 2016, scientists from Delhi University had taken Indian mustard, which was Varuna variety, and they have taken another East European mustard. First, they genetically modified these two varieties in order to cross-pollinate. After that, they cross-pollinated these two varieties, and finally, they got a new variety called as DMH11. Remember this one? What is DMH11? Dhara mustard hybrid 11. Normally, the mustard crop is a self-pollinating crop. As it is a self-pollinating crop, we cannot produce hybrid varieties in this one. So, in order to cross-pollinate them, in order to produce hybrid varieties, first the scientists have taken these two varieties, Indian mustard variety and East European varieties. First, they genetically modified these two in order to undergo cross-pollination. After that, these two were cross-pollinated, thereby they got a new variety called DMH11. And what is the benefits of this DMH11? So that means the crop production, the yield is 30% more than the normal variety. The crop production of this DMH11 is 30% more than the traditional mustard variety. The yield or the production is 30% more than the traditional variety. It also helps in boosting edible oil production. It boosts edible oil production. It also boosting edible oil production, and that too it is a herbicide resistant. So these are the benefits of DMH11. The crop production of this DMH11 is 30% more than the normal reference traditional variety, and it boosts the edible oil production. Okay, that is mustard oil production, and it is herbicide resistant or herbicide tolerant. Right? So these are some important examples of GM crops. So golden rice rich in beta carotene, flower sour tomato in which the gene have been suppressed to delay the ripening process. Okay, BT cotton and BT brinjal. So these are acting as bio-insecticides. Okay, and GM mustard. So hirudin gene have been introduced into that one. And then Arka Shubha is a marigold variety developed by IAHR, Indian Institute of Horticultural Research. Okay, the carotene content is very much high, that is double the normal value. 1.4% in normal what you call the marigold varieties, whereas in Arka-Subha it is 2.8%. Okay, that too the carotene can be extracted after, even after spoilage. And another one is HTBT cotton. HTBT cotton means herbicide tolerant BT cotton. Herbicide tolerant BT cotton. Okay, CP4 EPSPS gene have been introduced from Cyanobacterium agrobacterium tumefaciens. Thereby it develops herbicide tolerant. Okay, alright. Another one is DMH11. It is a hybrid variety developed by the scientists from Delhi University. Here Indian mustard variety and East European mustard variety have been cross pollinated. So thereby we got a new variety called as DMH11. Okay, so these are the benefits of DMH11. So the yield is, production is 30% more than the reference variety. It is boosting the edible oil production, and it is a herbicide resistant or herbicide tolerant. Got it? Right. And next one is biofertilizers. And the next one is biofertilizers. Biofertilizers. First of all, what are fertilizers? What are fertilizers? Fertilizers means eruvulu. Fertilizers means any substance which provides nutrient to the plants. Any substance which provides food material to the plants. That is called as a fertilizer. Here biofertilizer. Bio means living. Any living organism. Any living organism or its product. If any living organism, its product is used as fertilizer, then it is called as a biofertilizer. If any living organism or its product is used as a fertilizer, then it is called as a biofertilizer. Biofertilizer. Here, various microorganisms. The various microorganisms like bacteria, fungi, fungus, blue-green algae, some ferns, or you can say plants, and also some animals. Their products can be used as fertilizers. So the microorganisms like bacteria, fungus, blue-green algae, ferns. So these can be used as biofertilizers. So the bacteria like examples, you remember these examples important. The examples like rhizobium, the bacteria like rhizobium, azospirillum, azotobacter, these can be used as biofertilizers. The bacteria like rhizobium, azospirillum, azotobacter, these are used as biofertilizers. And the fungus like worm fungus, vesicular, arbuscular, mycorrhizae. Example, glomus. The specific example of worm fungus is glomus. You remember these terminologies. The fungus like worm fungus, vesicular, arbuscular, mycorrhizae. And then the blue-green algae like nostoc, anabina. We have to remember these examples. So you may get the question, which of the following is not a biofertilizer, or which of the following combination represents biofertilizer. Like that you will get the questions. So blue-green algae like nostoc and anabinum. This nostoc and anabinum. And the fern plants like azolla. So biofertilizers means if any living organism or its product is used as fertilizer, then it is called as a biofertilizer. Some important examples of these biofertilizers. The microorganisms like bacteria, fungus, blue-green algae and ferns or plants. So the bacteria like Rhizobium, Azotobacter, Azospirillum. Fungus, WAM fungus, vesicular arbuscular mycorrhiza. The specific example here is glomus. Then blue-green algae like nostoc and anabina. And fern plant like azolla. Okay, right. Let us see Rhizobium. You know it very well. Rhizobium, it is a bacterium. It lives as symbiont. In the root nodules of legume plants. Root nodules of legume plants like pulses. Rhizobium, it acts as symbiont or it lives as symbiont in the root nodules of legume plants. Example pulses. For example, if you consider the groundnut plant. So this is an example of groundnut. Example of pulses. If you consider the groundnut plant, on the roots, some bud-like structures or nodules are present. In these nodules, there is a bacterium called Rhizobium. This Rhizobium lives as symbiont. Symbiosis, I already told you. Symbiosis means mutual benefit. Okay. So two organisms are living together and both are benefiting here. Here the Rhizobium bacteria and legume plants are leading symbiotic association. Okay. Right. And this Rhizobium bacteria, it captures the atmospheric nitrogen. The Rhizobium bacteria captures atmospheric nitrogen and this nitrogen will be provided to the plant. Meanwhile, the bacteria will get food and shelter from the plants. So for this nitrogen fixation, the bacteria is having NIF genes, nitrogenase enzyme and leg hemoglobin. With the help of this NIF genes means nitrogen fixing genes, nitrogenase enzyme. This is nitrogen reducing enzyme, nitrogen reduction enzyme. So nitrogenase enzyme and leg hemoglobin. With the help of this, the Rhizobium bacteria captures the atmospheric nitrogen and it enriches the soil with nitrogen. It provides this nitrogen to the plants. So that's why the Rhizobium is using as biofertilizer. And also the bacterization process can be done with this Rhizobium. What is that bacteria? Bacterization. Bacterization means here this Rhizobium bacteria can be grown in the laboratory. The Rhizobium bacteria is grown in the laboratory. This Rhizobium bacteria is mixed with charcoal powder. Okay, this process, and it is mixed with legume seeds. So this process is called as bacterization. This is called bacterization. Just you remember the Rhizobium bacteria which captures atmospheric nitrogen, and this is acting as biofertilizer. Okay. In a similar manner, Azotobacter and Azospirillum, these are the free living nitrogen fixing bacteria. Azotobacter, Azospirillum, these are free living nitrogen fixing bacteria. And then what about VAM? VAM. VAM means vesicular arbuscular mycorrhiza. VAM. So here you remember this terminology, mycorrhiza. Mycorrhiza. You have to remember this terminology, mycorrhiza. What is mycorrhiza? It is symbiotic association between the fungus and the roots of higher plants. Roots of higher plants. You remember this one. Fungus and the roots of higher plants. This is important. So you may get the question, mycorrhiza is dash. Mycorrhiza, myco represents fungus, rhiza represents roots. Myco means fungus, rhiza means roots. Mycorrhiza. It is a symbiotic association between the roots of higher plants and the fungus. Here, suppose say this is a plant. These are the roots of these higher plants. And these roots are deeply penetrated into the soil. Okay, the roots are deeply penetrated into the soil. On these roots, the fungus grows. Fungus grows like this. Okay, and this fungus releases certain chemicals. So these are rocks. This fungus releases certain chemicals. The fungus releases certain chemicals. Okay, so these chemicals will dissolve the rocks. From these rocks, sulfur and phosphorus are formed. Generally, the rocks contain sulfur and phosphorus. Due to the action of these chemicals released by the fungus, the rocks dissolve, thereby they release the sulfur and phosphorus. And this phosphorus and sulfur they dissolves in the water, and this phosphorus and sulfur will be absorbed by the plants, absorbed by the roots. Like that, the fungus are providing sulfur and phosphorus to the plant. Meanwhile, the fungus will get the food and shelter from the plant. So that is both are mutually benefited. And this type of association is called as a mycorrhizae. Mycorrhizae. You have to remember that one. Okay. All right. And blue-green algae like nostoc and anabina. This nostoc and anabina generally they grows in the crop fields. They grows in the rice crops or wheat crops. They also enriches the soil with nitrogen. So nostoc and anabina, they also enriches the soil with nitrogen. So they can also be used as biofertilizers. And fern plants. Fern. The fern plant like azolla. This azolla grows along with anabina. The azolla grows along with anabina. So it can also be used as a biofertilizer. And also the various plant pots, the various plant debris, like leaves, fallen leaves, fruits, flowers, fallen leaves, flowers, fruits, and all these organic substances, that is humus, it can also be used as a fertilizer. The animals like earthworm, and also excreta of animals. This can also be used as biofertilizer. So the animals like earthworm, because these earthworms digs the soil, makes the soil porous, and also the earthworm excretes urea. Okay, from this earthworm, the manure is produced. It is called as a vermicompost. You remember this terminology, vermicompost. The manure prepared by using these earthworms is called as a vermicompost. So these are the biofertilizers. Then what are the advantages of these biofertilizers? What are the advantages of biofertilizers? Biofertilizers are eco-friendly. So these biofertilizers are eco-friendly because these are the organic compounds, so they are completely degraded. They won't leave any residues. They won't cause any pollution. Whereas chemical fertilizers, synthetic fertilizers, as they are synthetic in nature, they are not completely degraded. They leave some residues. They leave some waste material. They causes the pollution. Whereas the biofertilizers, as these are the organic substances, they are completely degradable. So they won't cause any pollution. So we can say them as eco-friendly. Okay. They won't harm the other microflora. They won't harm the microbiodiversity on the soil. Microbiodiversity in the soil. That means they won't cause any damage to the soil. And that too, the production cost of these biofertilizers is less when compared with the synthetic fertilizers. But the only drawback is storage problem. As these are the organic substances, they are easily degraded. So they cannot be mass produced. Unlike that of synthetic fertilizers. Are you getting? Arthamadunda? So biofertilizers are pollution free, eco-friendly antunam. Endukante they are easily degraded. Easy ga degrade అయిపోతున్నాయి. ఓకే, కాకపోతే ఒకటే డ్రాబ్యాక్ స్టోరేజ్ ప్రాబ్లం అనేది ఉంది. ఎందుకు అంటే బికాస్ దీస్ ఆర్ ఆర్గానిక్ సబ్స్టెన్సెస్, మనం ఎక్కువగా ప్రొడ్యూస్ చేసామనుకో అవి డిగ్రేడ్ అయిపోతాయి అన్నమాట. సింథటిక్ ఫర్టిలైజర్స్ అయితే మనం గోడౌన్లో స్టోర్ చేసుకోవచ్చు. ఇజ్ ఇట్ క్లియర్? దట్ ఈస్ ద డ్రాబ్యాక్. నెక్స్ట్ biopesticides. Another one is biopesticides. If any living organism or its product is used as pesticide or insecticide, any living or its product, any living organism or its product is used as pesticide, then it is called as a biopesticide. Biopesticides. Generally, these biopesticides may be of different types. They are biochemical pesticides, microbial pesticides, and plant incorporated pesticides. They are PIPs, plant incorporated pesticides. So biopesticides, these may be biochemical pesticides. Biochemical pesticides means generally they won't kill the pesticide, they won't kill the pests. Simply they interferes with their metabolism. Suppose insects are there. So these biochemical pesticides, these are the natural extracts from the plants. Simply they interferes with the metabolism of the insects. For example, insects. If you consider these insects, in these insects, the chemicals called pheromones are released. Pheromones. So these pheromones, they act as sex attractants. The pheromones are released, they act as sex attractants. So these biochemical pesticides, they interferes with the formation of these pheromones. When the pheromones are not formed, okay, that means the population, that means the copulation, the the potentiality of these insects will be reduced. So the population of the insects automatically decreases. So that means these biochemical pesticides, they won't kill the pests. Simply they interferes with their metabolism. They reduces their population by interfering with the sexual reproduction. Okay, these are called biochemical pesticides. And microbial pesticides means some microorganisms like bacteria, viruses, these are grown in the laboratory, they can be used as pesticides. Okay, say for example some microorganisms like BT, Bacillus thuringiensis. Okay, some viruses like NPV virus, CPV virus, nuclear polyhedrosis virus. Just you remember, this is for your understanding only. Okay, no need to remember that one. And then plant incorporated pesticides. Here BT gene, best example is BT gene. The BT gene have been introduced into the cotton crop. The BT gene has been introduced into the cotton, thereby it becomes BT cotton. That BT cotton is acting like an insecticide. That means the BT gene have been incorporated into the crop. So these are called plant incorporated pesticides. Here microbial pesticides means some microorganisms are grown in the laboratory, they are sprayed, they can be used as pesticides. Okay, right, these are the different varieties of biopesticides. Then what are the advantages of these biopesticides? Advantages. Usually they are eco-friendly, same as usual that of biopesticides, biofertilizers. They are eco-friendly. That means they are not causing any pollution. They are completely degradable. And some disadvantages also there. Some limitations are there. They are host specific. Or you can say target specific. High selectivity is there. And host specific. So the requirement of additional control measures, because these are all organic compounds now. Okay, so they require some additional control measures. Time of application is important. So it is knowledge intensive. The farmers need to be trained for the application of these biopesticides or bioinsecticides. So these are biopesticides. The best example is BT, Bacillus thuringiensis, Bacillus papilliae, Bacillus lentimorphus, Pseudomonas sporozoa. These are all different types of microorganisms which can be used as biopesticides. Then, so in order to promote the use of these biopesticides and biofertilizers, or in order to promote organic farming. What is organic farming? Organic farming means farming without the use of any synthetic pesticides or synthetic fertilizers. Okay, that is called as organic farming. Or the farming by using these biopesticides or biofertilizers, that is called as organic farming. In order to promote this organic farming or the utilization of biopesticide fertilizers, the government has established the NCOF, National Center of Organic Farming. National Center of Organic Farming at Ghaziabad, UP. It has 7 regional centers. So which are located at Hisar, Jabalpur, Nagpur, Bhubaneswar. Hisar, Jabalpur, Nagpur, Bhubaneswar, Imphal, Bengaluru, Coimbatore. So, NCOF, National Center of Organic Farming, have been established by the government at Ghaziabad, UP. It has 7 regional centers at Hisar, Jabalpur, Nagpur, Bhubaneswar, Imphal, Bengaluru, Coimbatore. And these regional centers have been in collaboration with regional nearby agricultural universities. Okay, in order to promote the utilization of these biofertilizers and biopesticides. And also government is initiating various schemes like Paramparagat Krishi Vikas Yojana PKVY. The schemes like Paramparagat Krishi Vikas Yojana, and RKVY, that is Rashtriya Krishi Vikas Yojana, Rashtriya Krishi Vikas Yojana. Next, NMOOP, that is National Mission on Oil Seeds and Oil Palm, and also ICAR schemes. ICAR, Indian Council of Agricultural Research. Indian Council of Agricultural Research. Indian Council of Agricultural Research. Next, biofuels. Right. So next one is biofuels. So what are biofuels? Generally, fuel means any substance which provides energy, that is called as fuel. Biofuels means generally these biofuels are produced from the living organisms or from their metabolic byproducts. So biofuels means fuels produced from living organisms, living beings, or from their metabolic byproducts. The fuels which are produced from the living beings or from their metabolic byproducts, they are called as biofuels. So generally the biofuels, that means they are produced from oil extracts. Mostly they are produced from the oil extracts like neem, mahua, castor, karanj. Karanj means it is pongamia, kanuga, pongamia, and jatropa. These are the major sources of these biofuels. The biofuels includes oil extracts from a variety of plants like neem, mahua, maduka, mahua. It is also called as maduka in India. Okay, mahua, castor, karanj, pongamia, and jatropa. So generally these biofuels, that is called as bioenergy. So overall usage of this overall bioenergy contributes nearly 10% of the total world energy demand. Biofuel contributes to nearly 10% of total world's energy demands. See, most of these biofuels are used for household purpose. Bioenergy usage, home, it is nearly 80%. Industry, it is near 18%. For transportation, it is only 2%. So there is bioenergy utilization. Utilization of this bioenergy in homes is 80%. In industry it is 18%. For transportation it is only 2%, very least percent. So here bioenergy means, biofuels means, if the fuels are, or if the energy is produced from the living organisms or from their byproducts, then it is called as a bioenergy. These biofuels contribute to nearly 10% of total energy demands of the world. So mainly the biofuels are bioenergies produced from oil extracts like neem, mahua, castor, karanj, jatropha. You remember these names. And utilization of this bioenergy for household purpose it is 80%, industrial purpose 18%, for transportation purpose only 2%. Then what are the advantages of this bioenergy? Advantages: these are renewable energy sources. These are renewable energy sources. They release relatively less carbon dioxide emissions than the fossil fuels. They are eco-friendly. They release relatively less carbon dioxide emission than the fossil fuels. So they are eco-friendly. They are renewable sources of energy. The renewable sources of energy, they release less carbon dioxide emission than the fossil fuels, and that too the production cost is low. The cost effective. Their production cost is low. Generally, these biofuels are extracted from the waste materials. Okay, oftenly they are extracted from municipal waste. So they involve in the pollution control process, because the biofuel extraction is mainly includes the municipal waste or waste materials. So they control the pollution. They control the pollution, and they also reduces dependence on foreign crude oil or foreign exchange. So these are the benefits of biofuels. Let us see some important examples of these biofuels. Bioethanol or biopetrol, biodiesel and biogas. So these are the some important examples of biofuels. Biopetrol means it is bioethanol. Bioethanol is called as a biopetrol. Mainly it is produced by fermentation process. Fermentation process. So bioethanol it is produced by fermentation process. Say for example, the microorganisms involved in the production of this ethanol by fermentation process. The molasses. So from this molasses the ethanol is produced. From sugar beets the ethanol is produced. That is called as bioethanol or biopetrol. And biodiesel. What is biodiesel? The biomethanol. Biomethanol is called as a biodiesel. Suppose if you consider this biopetrol and biodiesel, these are produced by transesterification process. Transesterification. You remember this terminology, transesterification. The biodiesel or biopetrol can be produced by this transesterification process. Transesterification. What is meant by transesterification? First of all, here you have to remember oil. So what is meant by oil? What is meant by oil or a fat? Generally, what is oil? It is triglyceride. Oil means triglyceride, or it is also called as triesters of fatty acids. Triglyceride or triesters of fatty acids. Triesters of fatty acids. So means what? This is a glycerol alcohol. This is for your understanding. Glycerol alcohol. It is esterified with 3 fatty acids. 3 fatty acids. Fatty acids. That means it is esterified with 3 fatty acids. Okay. This is called as a triglyceride. Triglyceride. If you observe the formula of this one, this is for your understanding only. This is for your understanding only. In exam point of view, just you have to remember this transesterification. So, what is transesterification? For your understanding, I am discussing this one. The glycerol alcohol is esterified with fatty acid, 3 fatty acids. That's why it is called triglyceride. Tri means 3. Okay, 3 ester bonds are formed here. One glycerol alcohol. If you observe this formula, CH2OH, CH2OH, CH2OH. This is glycerol. This is glycerol alcohol. Now it is esterified with 3 fatty acids. 3 fatty acids like this. This is esterified with 3 fatty acids. Fatty acid 1, fatty acid 2 and fatty acid 3. So like that 3 ester bonds are there here. Is it clear? So here generally it is esterified with glycerol. This glycerol is the simplest alcohol. It is having 3 hydroxyl group. So water content is more. Moisture content is more. Because of its moisture content, the efficiency of this oil will be reduced. Okay. So to increase the efficiency of this oil, this glycerol alcohol is replaced with either methyl alcohol. The glycerol alcohol is replaced with either methyl alcohol or with ethyl alcohol. Are you getting my point? Glycerol alcohol. Glycerol alcohol is the simplest alcohol. It is contain 3 hydroxyl groups. So because of the presence of 3 hydroxyl groups, because of the presence of more and more hydroxyl groups, the moisture content increases. If the moisture content increases, the efficiency of the oil will reduced. In order to increase the efficiency of this oil, we are transesterifying. Okay, we are allowing for transesterification with methyl alcohol or ethyl alcohol. When you add, suppose when you replace this glycerol alcohol with methyl alcohol, okay, this is methyl alcohol. Methyl alcohol, you will get methyl ester. Methyl ester. So in place of glycerol, if you add methyl alcohol, then you will get methyl ester, or it is called as methyl acetate. Methyl acetate. And this methyl acetate is called as a biodiesel. Biodiesel. Got it? Suppose when you replace this glycerol alcohol with ethyl alcohol, C2H5OH, this is ethyl alcohol, you will get ethyl ester. Ethyl ester, or you will get ethyl acetate. Ethyl acetate. And this ethyl acetate you called as biopetrol. Biopetrol. Got it? These points you have to remember. When you replace glycerol alcohol, with methyl alcohol, you will get methyl ester or methyl acetate. That methyl ester or methyl acetate is called as a biodiesel. Biodiesel. When you react this oil with ethyl alcohol, you will get ethyl ester or ethyl acetate. Ethyl ester or ethyl acetate. And this ethyl ester or ethyl acetate is called as a biopetrol. Biopetrol. Okay. So this biopetrol and biodiesels can be used as blending agents with fossil fuels. Fossil fuels. Okay. Right. So next, blending. Blending means simply it is mixing. Mixing. Say B5 blending. 5% biofuel is mixed with 95% fossil fuel. Fossil fuel. B10 blending, 10% biofuel mixed with 90% fossil fuel. And then B20 blending, 20% biofuel is mixed with 80% fossil fuel. 80% fossil fuel. That is called as biofuel. Blending. B5 blending means 5 liters of biofuel, 5 liters of biopetrol or biodiesel is mixed with 95% of normal petrol or normal diesel. So now we are following this B20 blending. B20 blending, 20% of biofuel, 20% of biopetrol is mixed with 80%. That is 20 liters of biopetrol is mixed with 80 liters of normal petrol. That is B20 blending. B20 blending. Right, next, biogas. Next, biogas. Biogas. So what is biogas? It is a methane rich fuel gas. Methane rich fuel gas. Methane rich fuel gas produced by anaerobic breakdown of waste, particularly animal waste. Anaerobic breakdown of actually biogas. What is biogas? What is biogas? It is a methane rich fuel gas. It contains 60 to 70% methane, that is CH4, and 30 to 40% carbon dioxide. Whereas the natural gas contains 80% of methane. Natural gas, write down here. Natural gas. So natural gas contains 80% methane, 20% carbon dioxide. Whereas the fuel gas contains, that is biogas contains 60 to 70% of methane and 30 to 40% of carbon dioxide. Okay. So this is called biogas. And this is produced by anaerobic breakdown. Anaerobic breakdown of animal waste. Anaerobic breakdown of animal waste. So animal waste by certain bacteria, methanogenic bacteria. So here methanogenic so the methanogenic bacteria are involved here. So what are those methanogenic bacteria? Methanococcus, Methanomonas. So the methanogenic bacteria like Methanococcus and Methanomonas, they involve in the anaerobic breakdown of this cow or animal dung. Suppose say this is a biogas plant. Say this is a biogas plant. Here the waste material is dumped here like this. It is compactly packed. Here this bacteria, they ferments this waste material, thereby they releases the methane gas and carbon dioxide gas, and this gas is collected through the pipes like this. So simply a biogas plant is constructed in which the cow dung or animal waste is dumped here and is compactly packed. Here the methanogenic bacteria like Methanococcus and Methanomonas, they degrades the waste material. Thereby the methane and carbon dioxide gases are formed here, and these gases will be collected for utilization. Is it clear? Right. These are the biofuels. Next one is bioremediation. right? Bioremediation. So what is bioremediation? So bioremediation means cleaning of environment by using living organisms. Okay, bioremediation means cleaning of environment with the help of living organisms. With the help of living organisms. Cleaning of environment with the help of living organisms. That is called as a bioremediation. So actually, some waste materials are decomposed by the bacteria and fungus. The natural decomposition process occurs. Natural degradation process occurs. Suppose when a dead body is present, for example, the bacteria and the fungus, they involves in the decomposition of the waste. They involves in decomposition. These are called natural decomposers. These are called natural decomposers or scavengers, or these are also called as replenishers. Replenishers. And also certain plants. Like mustard. Certain plants like mustard. They also involve in the absorption of some heavy metals. And this process is called as phytoremediation. Actually, bioremediation means cleaning of environment with the help of living organisms. Phytoremediation means exclusively plants involved in the remediation process. Then it is called phytoremediation. So for example, some mustard plants, mustard crops. The mustard plants absorbs the heavy metals. The mustard plant, it absorbs selenium. So that means it absorbs some selenium. And also another plant called, another fern called Azolla pinnata. Azolla pinnata. It absorbs the heavy metals like mercury, cadmium and lead. It absorbs the heavy metals like mercury, cadmium and lead. It metabolizes these heavy metals. So this Azolla pinnata can be grown specifically in the industrial areas to reduce soil pollution to some extent. This is called phytoremediation. Got it? So what is bioremediation? It is the cleaning of environment with the help of the living organism. So most of the bacteria and fungus, they involve in the natural cleaning process, natural decomposition process. They act as natural decomposers, scavengers or replenishers. And also certain plants like mustard plant, and also some fungus, some fern plants like Azolla pinnata, they absorb the heavy metals. And this process is called as phytoremediation. Phyto represents plants. So remediation means cleaning. So some plants are involving in the cleaning of the environment. That's why it is called as phytoremediation. Got it? And this bioremediation, if you observe the strategy of bioremediation, the bioremediation is of two types. One is in situ bioremediation. In situ bioremediation and ex situ bioremediation. What is in situ bioremediation? Remediation of the waste at its site. That is called as in situ. In situ means within the place. So remediation or cleaning of the waste at its site. cleaning of waste at its site, and exceeding bioremediation. So what is exceeding bioremediation? Cleaning of the waste outside its site. Outside its site. So here in situ bioremediation. In this in situ bioremediation, so it may be biostimulation, and bioaugmentation. Biostimulation and bioaugmentation. So what is biostimulation? Biostimulation. Biostimulation means, so various nutrients can be added externally to the growth of the microorganism in order to enhance the remediation process, in order to speed up the remediation process. So in order to speed up the remediation process, some nutrients can be added to the microorganisms. That you call as biostimulation. Are you getting? So naturally the remediation process is occurring. Suppose say here the remediation, natural remediation process is occurring here. Is it clear? So externally we'll add some nutrients. Externally we'll provide some conditions in order to increase the growth of the microorganisms, in order to speed up the remediation process. This is called as a biostimulation. Biostimulation. Got it? And this biostimulation, it may be of different types. Bio-sparging and bio-venting. Okay, just you remember biostimulation. And what about bio-augmentation? Augmentation means enhancement, development, you can say. Here, in bio-augmentation, some microorganisms are involved. So the microorganisms which involves in the bioremediation process can be grown artificially in the laboratory. They are genetically modified. Are you getting? Some microorganisms which involves in the remediation process can be grown in the laboratory and they can be genetically modified, and further they can be utilized for remediation process. For example, the microorganisms like oil jopper, oil vorus, Pseudomonas putida, GS15, Alga sarb. These are the examples of bioaugmentation. Oil jopper, oil vorus, Pseudomonas putida, GS15 and Alga sarp. These are the examples of bioaugmentation. What is bioaugmentation? Those microorganisms which involves in the remediation process can be grown artificially in the laboratory and they are genetically modified, and further they can be utilized for remediation process. And what is oil jopper and oil vorus Pseudomonas putida? So oil jopper, let us see oil jopper. It is a group of or mixture of microorganisms. It is a mixture of microorganisms which are used to treat oil spills or which are used to clean oil spills or to clear oil spills. What are oil spills? Suppose say this is a water body. Accidentally during transportation, if oil is leaked, then the oil forms a layer on the surface of water. This is called oil spill. Got it? The oil layer which is formed on the surface of water, it is called as an oil spill. So manually, that means the cleaning of these oil spills manually is difficult. It is a time-taking process. It is a cost. Okay, so the scientists have developed some microorganisms like oil jopper, oil vorus, and Pseudomonas putida. And these microorganisms, they degrades they degrades the hydrocarbon bonds of oils. Thereby the oils can easily degrade it. So oil spills can be cleaned by using oil vorus, oil japper and Pseudomonas putida. These are genetically modified group of microorganisms, okay, which can degrade the hydrocarbon bonds of the oils. Thereby the oils are degraded. Even this oil japper and oil vorus can be used in oil refineries. So they can be used in oil refineries to clear oil sludge. So what is oil sludge? The solid waste formed in the oil refineries is called as oil sludge. So nowadays the oil refineries like ONGC, Oil Natural Gas Corporation Limited, the oil refineries like ONGC, BPCL, Bharat Petroleum Corporation Limited, okay, HPCL, Hindustan Petroleum Corporation Limited, IOCL, Indian Oil Corporation Limited, RIL, Reliance Industries Limited. All these are various types of oil refineries. They are using oil japper and oil vorus to clean this oil sludge. What is oil sludge? The solid waste formed in the oil refineries is called as a oil sludge. Oil sludge. Is it clear? Right. And next, what about GS15? What about GS15? So GS15 is a genetically modified microorganisms by American scientist. GS15. It is a genetically modified microbe to clean uranium contaminated water. To clean uranium contamination. So this GS15 is a genetically modified microbe which converts the soluble uranium into insoluble uranium. Thereby the uranium can be filtered from the water. The uranium can be extracted or it is filtered from the water. So that means GS15 is a genetically modified microbe discovered by the American scientists, okay, which is used to clean uranium contaminated water. Next, algasorb. And next one is algasorb. It can absorb heavy metal ions from the wastewater or groundwater. It can absorb heavy metal ions from the wastewater or from the groundwater. It is an immobilized algae. Algasorb is an immobilized algae which can absorb the heavy metal ions from the wastewater or from the groundwater right. And next, these are the examples of in situ remediation and then ex situ remediation. Ex situ remediation. Ex situ bioremediation. Here we will follow various methods like land farming. Already told you, ex situ bioremediation means remediation of the waste at its outside the site. Land farming, composting, and bioreactors. These are the methods followed in ex situ remediation. Land farming. So contaminated soil is excavated and spread over the land. Here land farming means contaminated soil is excavated and spread over the land. This is called land farming. That means the contaminated soil is digged, excavated, and it is spread over the land. And it is periodically tilted for proper aeration. That is called as land farming. And next, composting. Composting. It combines contaminated soil with non-hazardous organic materials. Combining contaminated soil with non-hazardous organic matter. That is called as a composting. Next, bioreactors. Soil and water pumped up bioreactors. So bioreactors is an arrangement, or is it an equipment through which soil and water are pumped. Soil and water are pumped up from a contaminated plume and processed through an engineered containment system. Soil, that is a bioreactor, is an equipment through which soil and water are pumped. Okay, pumped up and processed through an engineered containment system. Processed through an engineered containment system. Right. So with that, I will conclude this biotechnology part. So next, I would like to start the space technology. Shall we have a small break? Okay, we'll have a small break, 5 minutes break, then we'll start.